Okay, hello everybody and welcome to this special session, to this panel. And today we have a very special guest. So I would like to introduce you Sir Richard Robert, that he is a Nobel Laureate in medicine. And I would like to ask some questions that you are interested about his professional life as a researcher, as a professional, as a person. So feel free. I don't know who would like to open the talk. So any question? So if not, I can ask him if you want.
No.
Okay.
Yeah,
Come on.
The young lady here has
yes.
a question. Yes.
What's your name?
So, hello, sir. Hello,
Hi.
ma'am. I'm Amuddin. I'm a second-year student in the School of Architecture, Planning and Design. I did some researches and I read a lot about your biography and your works and research. So the question that I would like to ask you is the following. So with the rapid evolution of molecular biology, how can the academic community stay ahead in preparing students for the future scientific breakthroughs?
Well, I think understanding the basics of molecular biology is important. And if you really have a good grasp on the basics, then you can easily follow the future developments. But ultimately, the key is always communication. And I think one of the problems that we find with many scientists is that they really don't communicate very well. They think that if they use technical terms, if they use convoluted language, shall we say, that everybody will think they're really smart. And especially they think their colleagues will think they're really smart. Whereas in fact, it's exactly the opposite. It's the people who can speak in language that everybody can understand that is important. I have something that I call the grandmother test that I give to my students. And I tell the students, you have to be able to go home, talk to your grandmother, and explain to her what you do in the lab every day, and explain in words that she can understand. And you can test whether or not she's understood what you've said, because she can then go and tell all her friends how smart you are and explain why you're so smart because of what you've been doing in the lab every day. And I think it's really important. It's something that I don't really care what field you're in, maybe in architecture too, this is important, to be able to explain to the general public what it is you do every day in words that they can understand.
Yes.
Yeah. Okay, do you have another question? Because according what Professor Robert explained us,
You can call me Rich. I don't like all this professor.
Okay, Rich, yes, you are right. We are between
call me
friends, in
Rich.
family.
Okay.
Rich said that communication is important, to be able to communicate the idea in which you are working in order to let all the people know what are you doing. But between you and the professor and the students or society, we have the media, we have the journalists. So what do you think about this intermediate? So because maybe sometimes they can manipulate information about the media. What do you think?
Yes, I'm not a big fan of the media.
Oh.
Because I think very often that what they do is they're looking to make a story that will captivate the audience and will allow them to continue working in the media. And for instance, in science, they very often do a very poor job, because if there are two sides to a story, then they will often say, oh, it's equal, 50-50. Whereas if you go to scientists and ask them about it, you usually find that 90%, 95% feel one way, and it's only a small minority who feel the other way. And yet the media never present it that way. Because if they presented it that way, then it's as though the decision has already been made as to what is correct and what isn't. And this was certainly something with climate change until relatively recently. They get the oil companies to come in and talk, and it's sort of 50-50. They look for the people who are skeptical. They still manage to find a few, but very few people these days are skeptical about climate change and the fact that we've caused it. You know, it's our problem. We did it. And unfortunately, the politicians are doing nothing about it, which is really very sad. And I think it's up to young people like you to speak out about this. You probably all know about Greta Thunberg in Sweden. Yes, have you heard of her?
Yeah, of course.
She's made a huge impact. You know, she's your age. She's young.
Yes.
You have the opportunity to change the world in ways that I don't. Okay, even though, I got a Nobel Prize and people listen to me. The world, the future of it is yours. It's in your hands. And if you don't speak up and make the adults behave properly, then you won't have a decent world to live in. So be activist. Very important.
So who would like to speak up now?
Oh, I'd like to go next.
Yes.
Thanks, ma'am. So, first of all, I'm Khadija Mkrim. I'm also an architecture student, but this time in the 4th year. So, my question to you, Mr. Richard.
Just Rich.
Let's go. Yeah, let's go like that.
Okay.
So, Rich, what ethical frameworks do you recommend that should be integrated into in the education of the new scientists, particularly those working on gene editing?
Okay. Well, so, gene editing is a very interesting technique that was discovered relatively recently within the last 10, 15 years. And it really has the great possibility of changing things dramatically. And there's one area that I'm especially interested in, which is the genetic modification of crops, of plants. Because one of the things that you may or may not be aware of is that, there are 800 million people who go to bed hungry every night because they don't have enough food. 800 million. There are children dying because they can't get enough food or they're missing key nutrients. And yet we can fix this. It's not an unfixable problem. We know that we can improve the yield of plants. We know that we can take, say, a small amount of space and double the food nutrition that we can get from it. We can improve the nutrition. There's something called golden rice. For an awful lot of kids, they go blind or they die before they're 2-3 years old because they don't get enough vitamin A. And yet there is a solution for this. For anybody who eats rice, there is a modified rice called golden rice, which now is commercially available in the Philippines. But it's been resisted by the anti-GMO people, Greenpeace in particular, because they see it's a nutrient. It's a pharmaceutical almost, and they're worried about that. So, there are things that we can do using these techniques, CRISPR techniques, in order to improve plants, and we need governments to change their policies and not to pretend that they're dangerous. And don't let the activists tell you they're dangerous, because they're not. They're simply not. We have 166 Nobel Laureates who've all signed on to say these are not dangerous. 166 of us have signed up and said they're not dangerous. There's not been a single case of a problem in the more than 30 years since they were first developed. People eat millions of tons of these crops every year. And the biggest hypocrites of all are the Europeans, right? So they won't import GMO crops because Greenpeace have convinced everybody they're dangerous. They're not dangerous, but they bring in millions of tons of GM crops to feed their animals. So the sheep, the cows, the horses in Europe, these are all eating GM crops. And apparently this is perfectly safe. If you're an animal, it's safe. But if you're human, it's not safe.
Yes.
I can tell you they're safe. These things are safe. And we should make sure that we do something to get this improved crops out into the hands of the poor people around the world who go to bed hungry all the time. Now, there's another use that people talk about from time to time, which is the use of GM methods in order to alter human beings. And you've probably read something about this. There was a case a few years ago of a Chinese scientist who deliberately made some alterations using CRISPR technologies in babies. And he spent 3 years in jail for doing that. And it was really stupid. He thought he was going to win a Nobel Prize for this. This is why he did it. He thought he was going to win a Nobel Prize. Instead, he made an absolute idiot of himself. And also, I think, made a lot of people think about what they wanted to do. Now, we know there are some things you can do. For instance, in the UK, there's recently been a technique to cure sickle cell anemia. Now, you probably know about sickle cell anemia here in Africa. It's quite a big problem for many of the population here. And you can fix this. And CRISPR can be used to do it. It's looking very good. It got approval in the UK. The only problem with it, it's unbelievably expensive. So you can't do it for more than a few wealthy people, which is sad. But I think, again, if the companies that sort of hold the patents on the CRISPR methods behave properly, they would not charge these enormous prices to do things. But we can also do many other things. We're going to be able to do them, maybe using CRISPR, maybe using whatever is the next generation that is better than CRISPR. But we are going to be able to do this. We will be able to alter the germline. We will be able to alter individuals. So if you happen to be Jewish and have Tay-Sachs disease, we can probably cure that before you're ever born. And there are many other genetic diseases we will also be able to treat. But I think it's important that we are very careful when we do it. We don't want to do it until we know for sure that it's going to work, that it's accurate, that it doesn't have side effects. And so we have to be extraordinarily careful. And I think we also have to be careful that there are not a few rich people who think it would be nice to have taller kids by altering their genes. You know, that makes no sense whatsoever to be doing that sort of thing. But for medical reasons, I think it can be very good. It's wonderful. And I also like the fact that the discovery was given the Nobel Prize to the two ladies who made the discovery. And this is something that's a little bit rare in the Nobel community. They don't give enough ladies credit for what they've done. Historically, this has been a problem. But at least in this case, the two ladies got it. Jennifer Doudna and Emmanuelle Charpentier. Very, very clever ladies.
So thank you, because as you said, if we don't know, or if we are not able to understand in a very clear way that this is not harm no one, this is very positive, not only to avoid hunger, to avoid different challenges that we are facing, maybe we are going to follow Greenpeace or this kind of institution that we think that they are the gurus of the nature. So, and if not, so we need more research, and we need to be more close to this kind of people that are researching a lot of years. So do you want to ask anything more about his professional life or another aspect that you are curious about?
I think the next two questions.
Okay, perfect.
And Mr. Rich, an architecture student in the same class as Khadija. Okay. And I'm curious to know about how can scientific communities better communicate with the skeptical public about the safety and the benefits of GMOs?
Well, I think there are several things that need to be done. So I go around the world talking about this. And we need to get more people who people will listen to talk about it. But I think if we could persuade the Europeans to behave properly and to stop listening to Greenpeace, that we would be 90% of the way towards having GM crops being just accepted as standard. If anything, they're probably safer than traditional crops. And it's crazy that the Europeans are opposed to this. And they're opposed to it for all the wrong reasons. They're opposed to it because Greenpeace, when they first came out, they said, oh, these could be dangerous. It was actually a fight against Monsanto. They didn't want the Americans to take over the food supply in Europe. And so they used this as an excuse. And they went out and they said, we need to do more science. Well, at the time, that was probably reasonable. Yes, why not? When a new technology comes along, let's do some more science and see if it's safe. But now we've done the science. We finished the science many years ago, and they're safe. These things are completely safe. So why doesn't Greenpeace admit it? They call for the science. Why don't they say, you guys listen to us, you did the science, they're okay. And you know why? Because being against GMOs was the best fundraising campaign that Greenpeace ever had. And so it's all about money.
So it's
If
business.
you ask, yes, if
behind.
you ask why people do things, 99% of the time it's because of money. They do it because they can make more money. And Greenpeace, because they were a non-profit organization and raising money, they could spend that money on advertising, on making movies to scare people, on getting you generally scared about all of this. The scientists, on the other hand, who were trying to defend themselves, you think when I apply for a grant and I say, well, I want some money to make a film to defend the science, we don't get money for that. And so it's as though we have our hands tied behind our backs trying to defend ourselves, and we can't do it. You simply cannot do it other than by talking to people. And so we've been trying to get Greenpeace to come forward and say, hey, you did the science, it works, these things are safe. They could say, hey, we've been right all along. But I'm afraid that they feel they're going to lose their fundraising if they do that, and they're not going to make as much money.
I think this is a great example of how a scientist or maybe a student, as you are, have to be very focused and have to be keeping on the looking for the truth and keep going. So I think it's a great example because I would like to know what is your motivation to keep going all the time. Maybe it can be like an inspiration for all the students. So motivation is important, and your motivation maybe is to fight against the poverty, the hunger.
Well, in my case, to make scientific discoveries.
Yeah, okay.
I get up every morning, I open my computer, I look at things that have gone on, because everything I do for me is on the computer. I make a discovery every day. You can't beat it. I don't think I've worked a day in my life. I just love what I do. It's my hobby. My wife
great.
doesn't always view it that way. She thinks I work, but I don't. I don't work. I just have fun making discoveries every day. It's great. You can't beat it.
It's great. Yes.
So maybe you should all think, maybe architecture isn't what you really want to do. Maybe you should head into biology and find something really interesting to do.
So any other question?
So, having fun making discoveries every day. Professor Rich was awarded the 1993 Nobel Prize due to your discovery. So I'd like to know, what are some lessons from your discovery of gene splicing and introns could be applied in current educational approaches in molecular biology?
Well, so I think it's actually quite interesting. You make a discovery, and sometimes it's very quickly converted into something useful, and CRISPR is a good example of that. It was very quickly, within a few years. I made the discovery in 1977, and it's only about 10 years ago that the first clinical application of this came out, when it was discovered by actually a fellow who was a postdoc with me many, many years ago, Adrian Craner. It was discovered that there was a defect in kids who had the disease spinal muscular atrophy. And it was because this process I discovered, RNA splicing, that enables genes to be processed within cells, was aberrant. Something had gone wrong with it, and he figured out how to fix it. And so now you have to take this medicine regularly, but now there are kids alive who would have died from spinal muscular atrophy because he followed through from the discovery that I made and was able to come up with a cure. And so I think, this is really what keeps you going, the fact that every time you make a discovery, I didn't know what it was going to be useful for. But, we couldn't have interpreted the human genome sequence when we had it if we didn't know about split genes and the fact that genes in humans were very different, the way they were organized from genes in bacteria. So, this is great. You hear about this, you find out what's going on. It's terrific. You can't bet it. And now there's, that's only one disease. There are other diseases that are aberrations in RNA splicing that can be fixed by similar kinds of techniques. And it's great. It's terrific.
Yeah. It sounds. Yes. So do you have more questions?
So I'm gonna ask. My name is Rania Salim. I'm a third year student in the School of Architecture, Planning and Design.
Okay.
And I was wondering if you can discuss a time when a cross-disciplinary approach in your research has led to unexpected insights or like even breakthroughs.
Interesting.
So let me talk about my own career and in terms of interdisciplinarity.
Yes.
So when I was a young boy, I loved mathematical puzzles. Okay, and I always thought I was going to be a professional mathematician. If when I was about 8 or 9, if you'd ask me, what are you going to do? I say, I'm going to be a mathematician. I love mathematics. I still do. I still love it. But then I read a book. It was called How I Became a Detective, a traditional detective, police detective. And by the time I finished that book, I figured this was what I wanted to do with my life. It sounded great, solving crimes, solving puzzles. It was sort of like the mathematics of puzzles and a use for that. And then my father bought me a chemistry set. And I discovered that I could make explosives, I could make fireworks, because these were the days when chemistry sets were actually interesting.
Yeah.
Nowadays they're pathetic, but these were really, and so I fell in love with chemistry. I then went on and did chemistry at university. I did a bachelor's degree and got a degree in chemistry, and then I went to do a PhD in organic chemistry. And I was lucky. I had a mentor who really, within 1 year, I had all the results I needed for my thesis. But just because of him, he told me how to do it. He really taught me how to do chemistry. And then I floundered around for a little while because you have to spend 3 years in England before they give you a PhD. And I used to go to the library and I read a book and discovered something called molecular biology. And I read another book by John Kendrew called The Thread of Life, which was all about the origins of molecular biology. It was sort of the origins of how chemistry led into molecular biology. And by the time I finished that book, I wanted to be a molecular biologist. And so when it came time to do a postdoc, I applied to 6 labs. Only one accepted me, one in the US, a man called Jack Strominger, who was in Wisconsin at the time. I took it, I only had one offer, so it was easy to make that decision. And so just before I was scheduled to go to Wisconsin, he wrote to me and he said, "Oh, can you delay coming for 2 months because I've just been made a full professor at Harvard." And so by chance, by luck, I ended up at Harvard and have been doing molecular biology ever since. So, I think reading outside of your subject, listening to talks outside of your subject, you can very often decide that there are things that are much more interesting that you can really get involved in that have nothing to do really with what you thought you were going to do, but then it can work out really well. And so once I became a molecular biologist, after we made our discovery of splicing, I became very interested in using computers to analyze sequences, because we first of all wanted to started sequencing things. We wanted to know what were the DNA sequences involved in splicing. And so we wrote many of the original programs to assemble DNA sequences. So, when you do DNA sequencing, you get little bits and pieces and you have to figure out how to put them together. I got involved in what is now bioinformatics, and bioinformatics is what I now do.
Wow.
But I'm starting to get very interested in artificial intelligence. And, maybe I'm going to switch and move into that area, because I think there's a lot of really interesting stuff going on there. So I like new stuff. I like to think about new stuff. And if you follow through on some of this, it can be great. Never be afraid to switch fields.
Yeah.
It really is quite important. If you find something you love to do, then do it. And bioinformatics solves everything I've ever been interested in doing. Mathematics is mathematical. Write computer programs. A detective, I'm solving mysteries all the time. Chemistry, molecular biology is all about chemistry. And here it all is in bioinformatics. So everything I've ever wanted to do all came together eventually. So it was good. It was good.
Great.
Yeah.
Okay. Okay, so do we have the last question?
Yes, I think I'll be the last.
Yes.
So, I have it, a third-year student in the School of Architecture, Planning and Design as well.
Okay.
And I'd like to ask a question about what we already talked about. So, what are the implications of alternative splicing discoveries for personalized medicine and the future treatment for genetic disease? Perfect.
So, I think the answer to that I've not covered already is interpreting the human genome. So you may remember many years ago, the human genome, we claimed that it was sequenced. It wasn't. I was actually chairman of the scientific advisory board of Celera, the company that did it. At that time, we had a pretty good idea of what the human genome sequenced, but it wasn't closed. It's really only within the last year that we've come up with a complete human genome sequence where we think nothing is missing. So that looks really good. Now, to interpret that sequence, it means you want to know what are the genes, where are the genes, what are they, and what do they do? And what one would like to do is to say, well, this gene does this. We look at someone who is ill, and we discover that there is something funny about this gene. And we can then make the case and eventually do the experiments to show that it is the defect in this gene that is the problem. Now, genes in humans are spread out. There's a little bit here, a little bit here, a little bit here, a little bit here. You make an RNA copy of everything, and then you take that RNA copy and you cut and splice it to join all of these little bits of the gene so that they become whole. It's exactly the way that a movie director makes a movie. You shoot a scene, and then you shoot another scene, then you shoot another scene, you go into the editing room and you cut it up, join it together so that when you go to the cinema, it looks as though it's one continuous piece of action. And that's how humans make the RNA, the messenger RNA, that is going to make the proteins that make us work. Now, sometimes this cutting and splicing goes wrong. Doesn't do it as it should because there's been a mutation in one of the points where splicing should take place. And when that doesn't happen, then it goes elsewhere and it makes a protein that is not correct.
Yes.
Now, if you can solve that, which is what was done for spinal muscular atrophy, if you can solve that, then you've solved the problem in a disease. And eventually one would like to be able to do that in the genome, back in the DNA that is being transcribed. But that's a long way off. We have a long way before we're going to do that on a regular basis. Maybe occasionally we can do it, but on a regular basis it's going to take a long time. But you have to know about all of this stuff before you can start to think about personalized medicine. We want to know what all of your genes are doing, and we want to know if one of your genes may be the BRCA gene, which makes you susceptible to breast cancer. Maybe if this is wrong, this is something we want to fix early on, find a cure for. And it's this kind of personalized medicine that can really be very helpful. And, this is what I love to do.
It's so interesting. We can see. So
Make me want to switch to biology.
Good.
maybe this is the real.
Apply to my lab, you can come and be an intern and we will work from there. How's that?
Yeah, that sounds a really interesting talk about ARN and DNA and so on.
No, it's really cool. Oh, it's all very cool stuff.
Yes, you have to have your eyes very opened in order to see new opportunities in which you can change maybe your way and maybe discover new opportunity. So I would like to thank you, all of you, for your amazing question. And of course, I would like to say thank you to Rich because your talk has been very, very, very inspirational. So thank you. And I would like to remind you in order to conclude. So remember that the curiosity is important and to be able to be flexible in order to change your mind and to keep going. So don't feel frustrated because it's not easy. But if you have a goal and you go directly to that, okay, that's perfect. But remember that in life we have serendipity, that this kind of things that happen by casuality, that maybe it's casuality, but I'm sure that there is another reason why so things happened. So, and I would like to finish my close, telling you, remember that when you have to lead yourself, you have to use your brain, your head. But when you are working in team and you have to lead others, you have to use your heart. Okay? So thank you for this time.
Thank you.
Yeah.
Thank you.